This is this is Group 4's Lab Report 1 on hand washing techniques. The purpose of this experiment is to see how effective washing our hands with soap is compared to when you're just washing it with water. It also helps us understand the importance of proper hand hygiene to reduce bacterial growth. Materials and methods. The materials used for this experiment include 4 blood agar plates for culturing the bacteria, a sterile cotton swab to collect samples from the thumb, saline to moisten the cotton swab, a soapy sponge for washing the thumb, and deionized water for rinsing the soap off the thumb. It's also important to ensure that all cotton swabs and saline solutions are sterile in order to avoid contamination. Procedure. Before beginning, be sure to label your 4 blood agar plates as shown above, R standing for right thumb and L standing for left thumb. Starting with your left thumb, first dip a sterile cotton swab in saline and swab your right thumb. Inoculate the section of the plate labeled R1. Next, in your workbench staining tray, rub your right thumb and pointer finger together under deionized water for 1 minute. Now inoculate the section of the plate labeled R2 with the sterile cotton swab. Then rub your right thumb and pointer finger under the water for an additional 2 minutes. Inoculate the section of the plate labeled R3 with the sterile cotton swab. Lastly, rub your right thumb and pointer finger under water for an additional 3 minutes. Inoculate the section of the plate labeled R1 with the sterile cotton swab. Now for your left thumb, start by dipping a sterile cotton swab in saline and swabbing your left thumb. Inoculate the section of the plate labeled R1. Next, in your workbench staining tray, scrub your left thumb with a soapy sponge for 1 minute. Rinse the soap off of the thumb with deionized water and inoculate the section of the plate labeled L2 with the sterile cotton swab. Then scrub your left thumb with a soapy sponge for an additional 2 minutes. Rinse the soap off of the thumb with deionized water and inoculate the section of the plate labeled L3 with the sterile cotton swab. And lastly, scrub your left thumb with the soapy sponge for an additional 3 minutes. Rinse the soap off of the thumb with deionized water and inoculate the section of the plate labeled L2 with the sterile cotton swab. These are the results for the left thumb. Starting with L1, in which no soap nor water was used, we can see a significant amount of bacterial growth on the blood agar plate. This indicates a high presence of bacteria before any washing. Next is L2, where we scrub the thumb with the soapy sponge for only 1 minute. As you can see, this effectively reduced the amount of bacteria. Then we have L3. Here we scrub the thumb with the soapy sponge for an additional 2 minutes, which completely eliminated the growth of bacteria. And lastly, we have L4, where we scrubbed for an additional 3 minutes, and this shows how effective that was because it led to no bacterial growth on the plate.
Hello, my name is Lucas Perez, and I will be taking over the next part of the presentation, which would be the rest of the results and conclusion. And to begin with, we have the results of the right thumb. So, in this part, it's the same thing that we did for the left thumb. We have 2 blood agar plates, left and right divided. And to begin it with, we have R1 on the left. On this one, even though we didn't have any water involved in it, we only had the saline solution to rub against the Q-tip with the finger, and on that gave us some growth signals in it. But the interesting thing is that on the second part of the right thumb, which is R2, we had higher growth than before. And what happened is that we found the presence of something called beta hemolysis. And it's often associated with S. pyogenesis, which is one of the bacteria that causes strep throat. But after further investigation, I found out that actually some of the skin flora can contain bacteria that can make beta hemolysis. And just to clarify, we have a lot of hemolysis. I think there's three types. We found out that it's beta hemolysis because it's clear. It has a clear area around the bacteria. And just for the record, this was 1 minute rubbing with water. Then R3 on the next plate shows us very little growth on it, like showing signals like kind of like the water is helping out, but at the same time it's not like fully doing its job. So we find that some microorganisms might be killed with just water, but after 2 minutes, if using water on the finger, we don't see that much of a change. And then lastly, on R4, almost everything is dead, like it's all gone. So this shows us that even though like water might take longer than water and soap, water can still like help. So if you don't have soap, it's way better to use water for a longer period of time than not washing your hands at all. Because with this, we do see the difference between washing and not washing our hands. So for this next section of results, we can actually see a pretty disappointing plate because unfortunately we didn't have any growth at all, or at least to the naked eye we couldn't see anything on the plate. And this was supposed to inoculate a swab from one of the bathroom door handles, but as I was mentioning, unfortunately didn't grow at all. So after investigating, I was like, whoa, like what happened? So I found out that probably my septic techniques weren't super correct and I need to improve them because otherwise we would have had some growth on it, or unless we have like a super weird bacteria that cannot be grown in like normal media. And and that leads me to like the thinking of maybe our bathroom door handles are really really clean, or my septic techniques were wrong, but I'm totally going towards the septic techniques being wrong, and we're gonna look forward to improve them throughout the semester. So for this last next section of results, we can actually see five plates, but I want to focus more on the four lower plates for now, which are the left and right thumbs. As you might be able to see, we have on the left the right thumbs and on the right the left thumbs. So this gives you a bigger overview slash perspective of how to compare and contrast when you use water to wash your hands or when you use water and soap to wash your hands. As you might be able to see, on the right side of the picture we have the left thumb that used soap. You can see how when we didn't use any water at all and you use the saline, you can see that it has a lot of growth in it. So you can see how by the next time, just a minute in water with soap, it's pretty much all gone. And then on the next one, it's also gone. And on the following, it's gone. So as long as you keep your hands, well, it's different for everyone. But in this case, we found out that washing your hands for 1 to even 3 minutes is going to help you out to contain the spread of bacteria throughout your body. On the other hand, we have the right thumb, on which we found that even though water by itself might not be as effective as water with soap, we found out that water in the long range can be more beneficial than don't washing your hands at all. So if you don't have soap close to you, like you cannot use soap to wash your hands, just remember that you have to wash for a longer period of time that you would have to do with soap. Because on this scenario, we see how non-water has growth, some water has also growth. Some water for a long period of time, it starts like decreasing the growth of bacteria, and after 3 minutes, most if not all the bacteria is gone. So by looking at this, I would give you like an eyeball result of like if you want to wash your hands with pure water, probably you have to do it around 3 to 5 minutes to kill all, if not all, the bacteria in your hands, which is way better than literally eating bacteria. So wash your hands.
Conclusion: as the washing time increased, the presence of bacteria decreased. This demonstrates the importance of thorough hand washing with soap to remove bacteria. These findings highlight the importance of practicing good hand hygiene to maintain cleanliness and prevent sickness.